The light excites the chlorophyll B's electrons in photosystem 2, which causes them to transfer and continue on to the next molecule in the electron transport chain, PQ. We can see that now photosystem 2 is missing electrons because the PG has continued them on in the electron transport chain. So the water molecule will split up, forming O2 and H plus ions, and the H plus ions, two of them will go to the photosystem 2 and replenish the electrons that were previously in the photosystem. Cytochrome complex, also known as the proton pump, will pump the protons against the concentration gradient using the energy gained from the electrons given away by photosystem 2. And the inside of the thylakoid will have a more acidic medium compared to the outside, resulting in a lower pH and a higher pOH. From the proton pump continues down the electron transfer state using a molecule called PC to transfer it to photosystem 1, where again using light from the sun, photosystem 1 gets excited and transports the electron to FD, which is another part of the electron transport chain, which is then transported to FNR, which is also known as NADP plus reductase. At FNR, it uses NADP plus and hydrogen ion from the environment to make NADPH, which is later used for the Calvin cycle. The 83 synthase now takes the hydrogen ion from inside the thylakoid lumen to the stroma. And then in the stroma, it phosphorylates ADP with another phosphate with the help of a hydrogen ion, causing ATP to be made.
The cytochrome complex, what it does is it takes these H plus ions and it pumps these guys through here because they're positively charged, and so they can't go through the bilipid membrane, or what's it called, phospholipid membrane, and so they have to go through the proton pump. And so what this does is they come through here. This increases the concentration gradient, which also in turn, what does it do? Let me see. Increases the concentration gradient and lowers pH. And lowers pH because there are more H plus ions over here now, like so, like this. Okay, cool. Okay, so now we have our electrons, and they continue down to this transport, and they go over here to photosystem 1. Okay, and what they do here is they continue up to the primary acceptor over here, and then they follow this acceptor over here, and then they follow this one, and they go down to the NADP plus reductase. Okay, so the electrons reach they go up here, NADP plus reductase. What happens is these guys go here, this plus this, boom, 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 boom. This creates this, boom. Okay, cool. This whole section is non-cyclic. However, as a result of the Calvin cycle, if we look at cyclic phosphorylation, then these guys, it would be the same. These guys would go up here, boom, 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 like this, to the primary acceptor. However, from the primary acceptor, there is this guy over here, another proton pump that can also transport these guys back over here so they can create more ATP. Cool. Thank you. Yeah. Okay, so this is the Calvin cycle. This occurs within the plant stroma, and it's light independent, meaning this can occur in the dark. Okay, so what happens is we have 3 CO2 molecules that come from the air, and we also have this RUBP molecule, which is a 5-chain carbon molecule. And using the enzyme Rubisco, we were able to carbon fixate this into 6 3-chain carbons, which is called phosphoglycerate. And using ATP, which becomes ADP, we were able to take the extra phosphate group and add it onto the phosphoglycerate. And what this results in is biphosphoglycerate. And so we have 6 of these right here. And during the reduction phase, this is the biphosphoglycerate is the intermediate compound, and in the reduction phase, it combines with the NADPH, which is produced in non-cyclic phosphorylation. And using this, when they combine, we produce G3P. The NADPH reduces down to NADP plus, and yeah, so we have 6 G3P molecules right here, and 1 G3P molecule will come out of the cycle, which can help create sugar in the future. And so, for example, we would need 2 G3P molecules to create glucose eventually. And this makes sense because if we're putting in 3 carbons, then we should be outputting 3 carbons as well. Using the resulting 5 3-carbon or 5 G3P molecules, we were able to regenerate RUBP using ATP to create RUBP again to once again continue this Calvin cycle. This is important because the Calvin cycle can create glucose, which can give more energy to the cell. And so in order to produce more glucose, the plant can go through cyclic and non-cyclic phosphorylation. And so if it were to go through non-cyclic or cyclic phosphorylation, then it would be able to produce more ATP, which could in turn produce more glucose for the molecule or cell.